Okay, can you
Hear me?
Excellent. Today's lecture is going to be on prions. This, again, because it is so weird. There is a fair chance that you haven't really heard of it before, but it really goes against the grain. So who has heard and roughly understand the nature of prions? Okay, that's cool. I may ask you later on a few questions. So I was in a couple of meetings, conferences and stuff throughout the 90s as a PhD student at Young Postdoc, when this kind of thing was, in full swing in terms of what was going on, why is prion disease so different from other diseases and so on. And I can tell you that the people who populated the field, I was not one of them, or anyone in our group was not one of them, was populated by weird characters. We used to say this must be infectious because just look at the researchers who work with it. So this was the most entertaining part of a virology conference, and I never missed a talk on prions. So therefore I'm a bit biased perhaps and thinks this is the bee's knees. So you can read a little bit in the textbook on this, but I will present you stuff on it that goes well past the textbook because I think it is so fundamental. Okay, that I'm not the only one who thinks this is the bee's knees is basically reflected by that the committee thought it fit to give 2 Nobel Prizes for one disease, which I think rarely, if ever, happens. So I hope it happens again in hepatitis C, but we see to this. So the first one in 1976 was given for the discovery of, for the discovery of new mechanisms for the origin and dissemination of infectious diseases. And the prize of that year was given to Baruch Blumberg, but that had actually nothing to do with prion diseases. He got this for hepatitis B related research. And the other one was given to Carlton Gajdusek for the description of the Kruger disease, and we go into this in some detail because in part is an Australian story, in the highlands of PNG. And this, by the way, but I will not make this the center of this talk, is one of the few Nobel laureates who made it into prison subsequently. So he is kind of famous/infamous for more than one reason, for whatever that is or amounts to. He is a very observant anthropologist, and he really was on the mark here early on. Then we have the 1990-70 prize for the discovery of prions, a new biological principle of infection, and the only prize holder is Stanley Prusiner, who I can tell you is a very confident man, and he, what they say in America, championed the idea to success. This is a backhanded compliment. That does not mean it's his idea. He just took it and ran with it and did this very successfully. Okay, that's why he gets in my little thing here on the black and white image. So then we have Charles Weismann, and he's also a bit of a character, very old school, Swiss. So he's dead now, but he gave excellent seminars. And he, I think, should have been awarded too, because his work with mice finally proved the theory of Prusiner and others. So yeah, but sometimes politics even works in the committee for giving away the Nobel Prize, and here I think it did. All right, so we start with PNG. And here's a map, and here is an insert, so it's a bit of a detailed area, and that depicts the area where the Kora people live. And they experienced an epidemic of quite a scale that was at times the most common cause of death for certain parts of the Fore population. And if you live in a jungle far away from any hospital or anything, this says something. So you're not shot, you're not clobbered, you're not poisoned. So this disease, this prion-related disease called Kuru, that was the most common cause of death. So Gajdusek, the first Nobel laureate, described several stages. The ambulant stage was characterized by an unsteadiness of stance, gait, voice, hands and eyes, deterioration of speech, tremor, shivering, loss of coordination in lower extremities that move slightly upwards, and dysarthria, slurring of speech. So it's all fairly unspecific. Then we have the sedentary stage. The patient can no longer walk without support. Everything is more severe. And there's emotional lability, outburst of laughter, which gave rise to the name of the disease, depression and mental slowing. And then there's the terminal stage, which we see here, marked by inability to sit without support, severe ataxia, tremor and dysarthria, urinary and fecal incontinence, difficulty swallowing, deep ulceration. So a terrible, terrible death. So death was, in any case, inevitable and usually occurred 3 to 6 months after the appearance of the first symptoms. And this occurred in a lot of people. But it's a story of the past. Local oral history taken when the disease was first studied by scientists in the 50s dated to an onset of the cases early in the 19th century. And it predominantly affected women and children. The children, however, of both sexes, with only 2% of cases in adult men. At the height of the epidemic, Kuru became the most common cause of death in women in affected villages. That says a lot if you live in the middle of a jungle in the 50s. Kuru was transmitted by cannibalism. Four communities practice the cannibalism of dead relatives as a mark of respect and mourning, so this is not like a consequence of tribal warfare or anything. Boys older than 6 to 8 years participated little in that type of mortuary feasting, which explains the differential age and sex incidence. So cannibalism was forbidden by the Australian government officers as one of the first acts of administrative control. After making contact with the Fora people in 1954, public consumption of the dead ceased almost immediately. Surreptitious cannibalism had been reported for some years afterwards, but at the end of the 50s, practice probably officially ended. More than 2,500 individuals died from Kuru. As far as numbers go, this is nothing if you compare it to, let's say, the AIDS pandemic, but this is a lot because these people were not very numerous, so a substantial amount of the population died of this. And here I actually have to kind of change this and show, because I just looked it up, and the last person was reported to have died in, 2009. So this thing was basically unraveled by Gajdusek, so who just observed how it happened. So he saw that only certain people got it and recorded the age and everything, and figured out that there was a link to the cannibalism here, and then they could basically make this go away by just stopping the practice. There is something to learn here, and one shouldn't be arrogant, because the West, in a way, failed to learn from this, as we will see in a minute. So, and it's probably on a upbeat note, is that Kuru is no longer diagnosed. So the region has a lot of problems, but Kuru is presently not one of them. Okay, so then it turned out that Kuru is not the only human prion disease. Kuru, sometimes called laughing sickness or shaking death, a disease that was once endemic in the Fora people living in the highlands of PNG, transmission by cannibalism. Creutzfeldt-Jakob disease, or CJD, known for a much longer time, but extremely rare. These occurs worldwide, and the incidence is 1 in 1 to 2 million. So you can be a GP and your son can be a GP and you never see any of those. So super rare. It usually affects people aged 45 to 75, most commonly appearing in people between the ages of 60 to 65. The disease may be inherited, that's called familial, arise spontaneously, that's called sporadic, or be caused through the transmission of animals or another human, and then it's an infectious variety. About 10% of cases are associated with certain mutations in the prion gene. So now we have for the first time like a link to culprit. And that is a gene that we all have, and which still is not quite clear what it does, but the fact that we all have it points to the fact that it's somewhat important, and that things can go wrong with the protein of that gene. Then there is another syndrome. It's called Gerstmann-Streusler-Scheinker, and that's a rare autosomal dominant inherited disease that strikes in the 3rd to the 7th decade of life, very similar to CJD, and that's associated with certain mutations in the prion gene, but slightly different mutations than the ones that give you CJD. And then there is a third problem that's called fatal familial insomnia, or FFI, a very rare autosomal dominant inherited disease that strikes in the 3rd to the 6th decade of life. The patient's progression into complete sleeplessness is untreatable, ultimately fatal. So, which also proves that you really need to sleep, associated with certain mutations in the prion gene. More recently, a variant, CJD, has been described, which occurs in younger people and might represent a zoonosis, in the widest definition of this word. In this case, a cattle-to-human transmission of prions. So, here we have the words again, fatal familial insomnia, Creutzfeldt-Jakob disease, Gerstmann-Streusler-Scheinker. So I think you do not need, you need to be aware that there are these diseases, but whether they affect the thalamus more than the cerebral cortex, this is the final point. So I think there's no need to learn all this. But all I'm saying is there's a group of diseases or syndromes associated with something going on with the product of the prion gene. And all these prion diseases are sometimes referred to as transmissible spongiform encephalopathies, or TCs. And to learn this is not bad because it A, shows you that this is transmissible, and then remember the Kourou case in PNG, yes. Then B, it has something to do with a sponge. And here we have brain tissue of a person who died of CJD, and we see the white spots here, and they are basically holes. And that is what gave the name to the, that's the reason why it's called TSCs. So they are spongiform lesions. What is also associated with all of those is the appearance of protein fibrils. These are these structures here. And they are made of prion protein, and they are made of misfolded prion protein. So you know that when a protein is made, it kind of flips into a particular shape, and that is then the shape the protein is active in. And if it's misfolded because something goes wrong in the misshaping, then these proteins are normally quickly degraded and disposed of and split into amino acids again. So here we have like large amounts of these prion proteins, and they organize themselves into these fibrils, and they are extremely resistant to degradation. And this is a problem. So then these things cause disease. And then, not surprisingly, it was found that you do not only have a couple of diseases in people, you also have a couple of diseases in animals. And scrapie, it's basically the same thing in sheep, However, so far there has been no evidence that if you eat an infected sheep that you can become infected too. There's chronic wasting disease in deer. There's bovine spongiform encephalopathy, or BSE, or sometimes mad cow disease in cattle. And this turned out to be like an epidemic worse than the one in Kourou as far as cattle are concerned in the UK. And then there is something transmissible mink encephalopathy in ranched minks, and there is something in cats as well. So here shows you a few photos in sheep, and the sheep, they also have like a set of symptoms that's not completely dissimilar to Kourou. It's not identical because we're talking here about sheep, but they are behavioral abnormalities. So they tend to scratch themselves to the point that they scratch all the wool off and even damage the skin. So they have the holes in the brain as, you know, their human counterparts. And if you look very carefully with an electron microscope, you find the fibrils as well. So they tick all the boxes for a TSE type disease. And they also have mutations in the prime proteins that kind of promote this. Then we have the chronic wasting disease in deer, and again, these deer show behavioral abnormalities, and it's also not, this one is already wasting, if you like. It's made to sin, didn't eat properly. So all not too dissimilar. Then we come to cattle, and that was a big deal in Europe. I'm sure it never reached Australia to that extent. But if you lived in the UK, it's probably fair to say that this to some degree was traumatic. So it started in the mid-80s, and then they realized that they had a spreading disease in cattle, introduced a feed ban, so some people weren't like completely stupid, but the introduction of the feed ban didn't actually initially do much to becoming more and more cattle affected. So I do not know how forcefully that ban was, monitored or what happened there, but so they really had a problem that reached well into the 90s. And during that epidemic in the UK, 200,000 diseased cattle, so there were confirmed cases, if you like, died or were killed, followed by a preemptive slaughter and destruction of nearly 4.5 million asymptomatic cattle. So they had not the facilities to get rid of them. So in cases they were just piled up on the field, oil, petrol, diesel pulled over them and just burned. So you may also think that was a bit over the top, but I can tell you, so you could drive through the countryside and there was no cattle alive. So that was truly traumatic if you were a farmer, I think. And it was economically important, all of this. So there's also lessons to be learned, here. I like to give a strange kind of credit to the British Minister for Agriculture, Conservative. The name John Gummer, we see him here, chomping into a hamburger, and he tried to feed his daughter one too. The daughter actually on camera refused to eat it, so he just ate both. And to promote the fact that British beef is safe. That was probably a bit premature, because what we have here is confirmed clinical cases of bovine, it's another word for cow or cattle, spongiform encephalopathy. So it's basically, I showed you that before, so it's the kind of graph that kind of starts in the mid-80s and goes well past the 90s. So what we see here is the cases of a new variant of Creutzfeldt-Jakob disease. So that started in the mid-90s, and to some degree it may still be ongoing, so I don't know what the latest numbers are. But from an epidemiological point of view, this is not causation, it's correlation. It strangely appeared after the peak of this. And what we know from the Kourou case and other cases, that incubation periods for this type of thing can be quite long. So when they banned cannibalism or convinced people not to do this anymore in the 50s, and the last person died in 2009, and given that this was not likely a spontaneous case, then that means there could be a 50-year incubation period. So this thing may have a tail. So, and why they think that this is something new, and not just because after this, you can imagine a lot of money was spent for prion-like diseases in the 80s and 90s because they really had a problem in the UK. Then they would also look very heavily in every old person who died and may have found cases that previously were overlooked. So this is always a bit dangerous if you do little epidemiological studies like this. However, these cases have something that's called a florid plaque pattern. You see, like this, almost, someone was inspired to say, oh, that looks like a little flower, rather than a simple hole. So that differs from classic Creutzfeldt-Jakob diseases, so therefore they think this did not happen before, and therefore they think this is a consequence of that. So, yeah, then given all this may not be as safe as it seems. Okay, so does it have anything to do with Australia? Australia has never had a case of BSE and has in fact been categorized as one of the least likely countries in the world to ever find a native bred animal with the disease. The position is largely based on the fact that no meat and bone meal has been imported into Australia from any country other than New Zealand since 1966, and also that we keep a lot of our farm animals largely dispersed, widely dispersed in a big country. So, yes, so we do not really face a problem of new variant of Creutzfeldt-Jakob disease as a consequence of diseased cattle. Then the other problem is migration, particularly with the incubation period of up to 50 years. So therefore, Australia has a policy of deferring blood donors who have lived in the UK for longer than 6 months between 1980 and 1996. And this has been instituted to reduce the theoretical list of transmitting the variant Creutzfeldt-Jakob disease through the blood supply. This is not as exotic as you think. It works best, the human-to-human transmission, if you eat someone's brain, because they're the most prion proteins. However, there are prion contaminations in other parts of the body as well. So there were cases in the past, that was before recombinant human growth hormone was used to improve the situation for genetically stunted people, so they get to what's considered a normal height. And previously that was that growth hormone was retrieved from cadavers, so which you may think is the medical way of cannibalism in a way. So, and if that happened from a person who had prion proteins, then disaster. So with recombinant human growth hormone, does not happen. So therefore, with all these experiences, we monitor still the development of variant Creutzfeldt-Jakob diseases and the transmission, because this has consequences, as we see here. Okay, so now we come for what's actually behind all this. So, and I told you that the concept of champion someone else's idea may get you a Nobel Prize if you're really good at it. In this case, it goes back to the 1960s, where a radiation biologist called Tikva Alper, that's this one here, and the mathematician John Griffiths, so that one, developed their hypothesis that TSEs are caused solely by proteins. They were, I think, not even proper biochemists or anything, so they were rather mathematicians or statisticians. The theory was developed to explain the discovery that the mysterious infectious agent causing Scrapie and Creutzfeldt-Jakob disease was resistant to UV radiation. This is important. So you know firsthand, basically, from the practicals, that you can kill bacteria with UV radiation. I told you that you can kill the bacteria because you damage the nucleic acids. And you can damage with UV radiation nucleic acids anywhere. You can do this yourself if you go to the sun in the midst of summer and, prepare yourself for a tan and a subsequent cancer. And then you can kill yourself that way. So, and you can use the UV radiation in the lab to sterilize like special equipment by putting heavy UV on it. So, but they found that the prion agent that could transmit the prion disease from one cattle to the next or from one person to the next was more or less resistant to UV radiation. So they thought like, oh, this is strange. There seems to be no nucleic acid, so no RNA, no DNA. And so what is actually causing the disease if you have no carrier of genetic information? So, and this was basically developed early on in the game by these guys, and they published that in a series of papers in, 2 of them in Nature. So it's not like, some obscure stuff, may have been overlooked for 30 years. No, this was very prominent. Okay, you can also read the bibliography of T. alpa, which is, for various reasons, very interesting. Okay, so then, Prusiner, he basically gave the whole thing a very catchy name, which is a bit of a trick if you want to be famous. So he coined the word prion, and that's a combination of the first 2 syllables from proteinaceous and infectious. So an infectious protein, and since this goes a little bit against the grain, because normally we think we have bacteria and viruses and parasites, and they all have nucleic acids, but here we have a protein that itself can make you sick and is infectious. So, and then your own protein, which you make all the time, which we make all the time as we speak, is the prion protein C, and C stands for cellular. And to distinguish that from the prion protein that makes you sick is then the PRPSC, stands for the prion protein in that pathogenic form and conformation. Here's a model of the human prion protein. It's a membrane protein. People still argue what it actually does. So there is some idea that it can make mice smarter. And if you knock it out, they are a little dumber or can't remember things so well. So, but I don't know whether this applies to us as well. So there must be some function. However, these prion proteins can go from one conformation into the other. And if this happens, that is doomsday. So this is a normal form of the prion proteins, called prion protein C for cellular, and this is the abnormal form, SC for Scrapie, and you see you have fewer alpha helices and more of the beta sheet structure. And if a protein goes from this kind of structure to this kind of structure, if I look at it and say, oh, this kind of structure looks like they want to stick together more. So the beta sheets like each other a lot, usually. And this is indeed the case in here, and this one aggregates with itself efficiently and forms these fibrils. Okay, and this can lead to a chain reaction, and that is basically the problem. So here, in gray, we have the prion protein C that we all have as we speak, and if someone is so unfortunate that 1 or 2 or 3 of them flips into the wrong conformation, and then they stick together, and then they can convince others very easily to flip into the wrong conformation as well, then basically the wrong conformation ones grow in numbers. And then they make these fibrils, and then you get sick. So this can happen, bad news, at any given time, and you do not know, and you may not know after 10 further years, but I tell you, it's very rare. Remember Creutzfeldt-Jakob disease, 1 case in 1 or 2 million people, and the other FFI and so on, they are similarly rare. So it's not likely that it will happen. Okay, so how did they find out that this is indeed the case? Now we go back to Charles. So he published a paper in 1992 in Nature, so here, that's him, about the normal development and behavior of mice lacking the neuronal cell surface PRP protein. So he made knockout mice. Are you familiar with the term knockout? Who is not familiar? Because then I will explain it. Okay. A knockout animal or plant is one in which usually a gene has been damaged or removed, so they do not make the protein that is usually encoded by that gene, and so the gene function is knocked out. And that's basically by genetic manipulation. There are several ways of doing it, but the result is always the same. They usually have one protein less. Not a single protein molecule, I mean one type of protein less. So in this case, they do not have the prime protein. And he got a Nature paper. Normally, you do not get a Nature paper for just making that knockout mouse strain. But because everyone was waiting for it, and they thought, like, if you knock out the prime protein, then, I don't know, you will have a monster, or the mice are not vital, or something like this. And then the mice appeared superficially absolutely normal. So, and that was newsworthy to be published in Nature. But that was followed, and you see 5 years later, not these people were slack or did not have any funding, because the development of the disease takes so long, by a paper in Cell, which is even more prestigious than Nature, at least as far as the impact factor goes, and was probably even the Cell cover, was mice devoiding of the PRP are resistant to scrapie. So if you do not have the PRP, you may be a little dumber, but you will never get scrapie or any prion disease. So, and that really proved it, that this gene and probably nothing else is critical. But Charles is Swiss, and my PhD supervisor was also Swiss. These two were actually close. And they make you do the same experiment over and over, you wouldn't believe. So that's why I'm saying it. Here he proved, and this, by the way, is a Kaplan-Meier plot. You start, let's say, with 50 animals, and you do something to them, and then you see how many animals are alive after a day, a week, a month, a year. So what we see here in green, and you cannot see the green dots here initially, is that mice, these knockout mice with no prion protein at all, they were infected, and so they were given infectious prion proteins, and then people waited what happened. And after 200 days, nothing happened because they're all still alive. So they could not be infected, they were resistant to this disease. So normal mice with the normal mouse prion protein, they started to become sick after, 120, 130 days, and all of them were dead after 150 days. So mice have a much longer life expectancy as humans, so this would translate into years if you would just go on into the lifespan of humans, or decades even. So this is a normal thing. This is what we would expect. This is the proof that if you knock out the prion protein gene, then the mice are completely resistant, and then they, trying to better this, even said, look, okay, then we make a transgene and give the mice their prion gene back, but with a stronger promoter, so they make more of it. Not the normal amount, so they make, I don't know, 3 to 4 times more of it, or 6 to 7 times more of it. And interestingly, if you infect these mice get sicker quicker. And this, again, kind of nicely shows it, so they call it kind of a dose-response thing. And that really did it in terms of settling once and for all, what is mainly responsible for this whole group of diseases. So that's why the Nobel Prize was not given before this was clear. Unfortunately, this was not given to Charles. All right, but Charles being Charles, he didn't stop there. So then he said, like, okay, we have mice with the mouse prion protein, and since we also have mice with no prion protein, we can now make them transgenic, not again for the mouse prion protein, but for prion proteins from other species. And here they used hamsters. So they made mice that are mice for all aspects, but for the prion protein, because the prion protein these guys make is the prion protein of hamsters. And normally is a little difficult for the TSE or the Kuo-like diseases to go from one species to the other. So that's why we do not get sick if we eat sick sheep. But we do get sick if we eat sick people. And we may get sick if we eat sick cattle. Oh, it's called beef. So there is a bit of, and this probably relates to the fact that some prion proteins from different species kind of fit together quite nicely and others don't fit at all, and if they don't fit at all, you are resistant. So to prove this, he gave the mice the hamster prion proteins, and voila, they were susceptible for hamster prion diseases after this. So that again explains, why there is that strange species barrier. So now, in the end, I would like to come to Creutzfeldt-Jakob diagnosis. I don't think you need to learn, the particular details of what kind of tissue you can retrieve to do a diagnosis with what kind of test. That goes a little bit too far. So important probably, if the person is dead and you can do an autopsy, then you can cut into the brain and look for fibrils and holes and all this, and this is the classic way of doing it. But if the person isn't dead, then you cannot obviously do this because that will impact the patient. So people developed some sort of weird PCR-like assays. And there are two competing ones. One is called RT-QuIC, and the other one is abbreviated PMCAs. They're very similar, so I have a nice diagram here from the protein misfolding cycling amplification assay, or PMCA. And what it is you take the brain, and then you make you break it up and use that as a source for cellular prion proteins, PIPC. And then you have a test sample that comes from an animal or person you suspect of having misfolded prions. So, and you use this as a seed and give it basically more prion proteins, and then you see whether you get more fibrils. And because instantly it's not very efficient, you get a little bit more fibrils, or you have already fibrils, and then they get a little longer, and then you're stuck measuring this, and it will be very difficult. So what you do is you incubate those, and then you use a sonicator. So you basically break everything up, and then you give it more of the prion protein C substrates, that's depicted here, and then you grow more fibrils. And then you use again the sonicator, and then do again, and then you have cycles where you amplify the fibrils. And if there weren't any fibrils here, then after a couple of rounds, there will still be no fibrils. But if there were a few fibrils, and so few that you couldn't even see them, but after a couple of rounds, you may have so many of them that you can easily see them. So this is a kind of test that will amplify the signal and tell you whether this test, this test sample is positive or negative for prion proteins. So they do not actually do microscopy. They take advantage of the fact that the fibrils, the prion proteins in the fibrils, is extremely resistant to proteases. These are enzymes that cut proteins. Whereas the properly folded prion protein can be degraded. And then you can do a Western blot. So that's a technique to have a band for a protein. And if you can not digest your fibrils, then they will form a nice band here. So that's basically the readout. So this is, I'm not saying this is a PCR, but in a way, this kind of cycling and adding building blocks is PCR-like. Okay, this is shows the same thing again, what I tried to explain using that flow chart here. And I like to show it again because it's again the principle of the whole thing. So here we have the misfolded prion protein. They form these aggregates. Then we have the normally folded prion proteins. They're kind of individual, but if you put them together, then the aggregates recruit the normal ones, force them into the wrong conformation, and then they recruit more. And the sonification, that's these symbols here, that break that up, and then they act as seeds to make more and bigger complexes, and then eventually you reduce the number of this, the properly formed prime proteins, and you increase the number of the, sick-making, improperly abnormal prime proteins. Okay, and this is the last slide, and this is, no pun intended, food for thought. Cannibalism rife among prehistoric humans. So, some geneticists analyzed the DNA from some Fora woman, that was actually done years ago, who had participated in many cannibal feasts before they were banned, but did not become, a victim of this disease. So they thought like, oh, maybe something slightly different with those compared to the people who became sick and died. And then they found that the survivors of the Kuru epidemic had a particular genetic signature. They were heterozygous for methionine at codon 129 in that prime protein. So there's no need to remember that this is the position and this particular amino acid. What is probably worthwhile is that these were heterozygous. They were not this, they were that. They had two copies, and one had methionine and the other one did not. And this is a bit unusual. So in support to this assumption, none of the patients who have contracted the variant Creutzfeldt-Jakob disease in Britain carried the protective signature. And usually, natural selection will favor one version of a gene over another. The favoring of multiple alleles together is called a balancing selection. It's rather rare, but it's the case here. So now people speculated that something favors the presence of both alleles in the human population. So there must be an advantage for both. And they think there's an advantage for actually being heterozygous. And then people thought like, oh, maybe every, and then people develop the tendency to eat each other. And this is not like last year, so this is kind of 1,000 years ago, 10,000 years ago, 100,000 years ago. And then this may, come down and will be banned eventually by social norms, but may leave a trace in the genetic makeup of people who run around today. So one shouldn't look down on people in PNG because you may be the descendant of people who did exactly the same thing. So on the other hand, it's a pretty good idea to be very careful to form these closed cycles. So the whole idea in the UK was that if you slaughter a cattle, and then there are things you eat, and there are things no one wants to eat, and then they autoclaved it and gave the protein back to other cattle, so they grow faster. So, and then, as far as I understand, there was a time when the regulations or whatever, the way the food was processed before they did it was changed, and the prime proteins are also quite resistant to heat, so their normal autoclaving doesn't really destroy the infectivity as it would with something that is actually truly alive, like a bacterium. So, after that was changed, the cattle in the UK were fed infectious material. And where does the infectious material come from? If you have millions of cattle and every now and then one flips spontaneously, which is normally not a problem because cattle do not eat cattle and then it would all die out. But if this one gets processed in little food pellets and distributed to 500 cattle, and then 100 of those come down the disease, and then you process the remnants again, because you may not notice, into food pellets, and then, boom, exponential. So that is what happened in the UK. So now people are much, much more careful of feeding the remnants of one species to the very same species in agriculture. So, and it doesn't really matter for this, probably the leftover of salmon, feed to salmon again would be a similar story. Not that I would be very afraid of getting prion-like disease by eating infectious salmon because the salmon is so far away from us in a relationship, but you may have traumatic effects on the salmon industry, which you may, for different reasons, also not want. So generally speaking, cannibalism, or the equivalent in animals, is not advised. So you can feed the salmon to the chicken, you will probably safe. You can feed the chicken to the salmon, it's also safe. But chicken to chicken and salmon to salmon and cattle to cattle and people to people is not safe. Okay, so that's my excursion into prions. Learning objectives, questions?
In that study where they did on the Kura people at the end there, that last slide, did they also do the genetic testing on the male population at any point to see if the heterozygousness was gender dependent?
I don't know, because I did not read the original publication. As a good, if this would be a study in animal, you would easily do it. A study in people, requires ethics clearance and blah, blah, blah. So therefore the numbers are never that high, but they may have done it. And it's likely even, but I don't know.
Do they have a figure? Is it the incubation period for these prion diseases being decades?
Yeah.
Is that due to the slow growth nature of neural tissue? Like there's only so many prion proteins that are produced in the brain over time, so it takes it's a longer loading period for there to be enough for an effect to.
No.
Do they have an idea of why?
I think that the initial stages, so it's exponential, the conversion, it's a chain reaction, so the initial stages are very, very slow. And it's a progressive disease, so the further you go into the stage, the faster it accelerates. So that's one thing, but apart from all this, I'm sure that I remember there was a dose dependency. So let's say you eat something that is not from your own species and you eat only little of it, then you start with a very low dose. So then you go from 1 to 2 to 4, this kind of thing takes a long time. But if you eat the brain, an infectious brain, late stage of your own species, you may start with a much higher dose. And you basically cut a little bit to the chase, so then it will be faster. So then, as it seems in that very last slide, that certain mutations make it easier for the proteins to flip in the wrong. Then if you have those and then get seeds, you do not wait until they flip spontaneously, but if you then help it along by having a large amount of, because you get a human growth hormone that was isolated from a infected person, then it probably also goes rather quickly. Any more questions? So there's no one waiting outside, so we do not have to evacuate in a hurry. Okay, then I will see you soon for the next round of lab classes.